Okay, last video. Gonna try and get it into 3 videos. All right, your deep thought question is why is gram-negative bacteria colorless, but a gram-positive bacteria purple after it is rinsed with decolorizer? Think about it, pause it. Remember, because the ethanol and acetate mixture of the decolorizer disrupts that outer layer in the gram-negative, and the thin peptidoglycan that's in between doesn't absorb. So there's no outside peptidoglycan in the gram-negative. So it doesn't absorb the purple. Then when you wash the purple away, the primary dye washes out because it's not grabbed on, because there's no peptidoglycan for it to attach to. And then they're colorless. So you will have purple and colorless. The thicker wall of that gram-positive bacteria is unaffected by the decolorizer because it has really absorbed it, and you can decolorize it all day long and it's like, whatever, this is the color I'm staying. So the purple stays because it's attached to the peptidoglycan, and the gram negative doesn't have any peptidoglycan to attach to. So it becomes colorless until you add the safranin, which makes it pink. Okay, we're talking a little bit about taxonomy, but only in ways in which we can find our unknowns, right? Not just your unknown project, but like, again, you're somewhere and you're trying to figure out what is causing everyone to be sick, and you have a microscope and some dyes. So Linnaeus, yes, we know, yes, domain kingdom, rights, surely this is not new information. So we're going to kind of skip over that. Animals, plants, fungi, protista, prokaryotes. We are in the prokaryote. The domain, eukaryote, bacteria, and archaea. Okay. Physical characteristics, biochemical tests. That's what we're down to. So physical characteristics are like, it's a bacteria. I don't know what kind it is, right? That's where we've gotten to. Hey, I'm looking at this, and I feel like it's a bacteria. Okay. They sort of have a distinct appearance, right? So, okay, we figured out it's a bacteria. Now what are we going to do? We're going to do some gram stains, right? Let's say we're going to do some gram stains. We're going to distinguish whether it's a prokaryote. We're going to do some biochem tests. This is what we're going to do after we do staining. We're going to do a bunch of these kinds of tests. Possible. One of the things, right, when you start narrowing down your unknown and when you start thinking about what your bacteria might be, it's going to say, does it produce gas? So what that means is, are there little gas bubbles in it, right? So we're going to put inverted tubes in here, and we're going to see if your bacteria produces gas. Does it produce hydrogen sulfide? These are all the kinds of tests that we can do. You can do these rapid tests. This is obviously what you're going to have like automated in a hospital somewhere when you need really rapid. And it's going to tell you, oh, does it do glucose? Does it do citrate? Does it do some of these things? There are serological tests. There are antibody antigen tests that you can do in a hospital. Again, antibody antigen. So if it has a specific antigen, it will agglutinate. That's the same thing if you do a blood test and you see it like coagulate, kind of become a solid. So in this case, you have a negative result. In this case, your antigen bound with your antigen bound with your antibody, and you get this test. And that's something, we are not going to do that one, but that is possible. You can do phage typing if you have a bacteriophage. Looks like that. You can look at nucleic acids. We are not going to do that one, but it's possible. You are going to use a series of dichotomous keys. So essentially, you're going to say, is it gram-positive? Yes or no? Let's say, no. It's not gram-positive. It's pink. I have rod-shaped cells. Okay, I have rod-shaped gram-negative cells. Can it tolerate oxygen? Okay, obviously, it tolerates oxygen because, dude, you're growing it in the lab. You're not in a non-oxidated environment. Can it ferment lactose? We will do a lactose test. Yes, it can ferment lactose. Can it use citric acid as a carbon source? We have a citric acid test. Yes. Can it produce gas from glucose? Yes. Then you have E. coli probably, or maybe you have Citrobacter if it produces hydrogen gas. Okay, these are how we're going to do the lab. Again, dichotomous keys. These are all the things that it will do. It will move you through something like this. So is it gram-positive? Yes or no? Is it rod-shaped? It could be. So really, if you're gram-positive, yes, and it's rod-shaped, then you probably have Listeria. Boom, done. I'm not giving you Listeria because it's too easy. Do you have Neisseria? Can it tolerate oxygen, right? These are the dichotomous keys that we're going to use and think about and even make some in class when we get to specific bacteria. Right? And here's the tests that we will do. Does it ferment lactose? Yes. Can it do this? Yes. See how this ferments? This is the gas bubble right here. Why did Linnaeus not create taxonomy groups for viruses? Come on, this is an easy one. This is an easy one. Because there weren't any viruses. I mean, there were viruses, but Linnaeus didn't know what a virus was. So that's why. Okay, nope, that's it. Okay, so we got 2.5 videos. All right, bye.